My name is Dr. Chris Thompson. Thank you for coming to my channel. I'm a professor of neuroscience at Virginia Tech, and I am starting a new series where I am talking with scientists doing really cool, interesting work. Just to kick us off, I've reached out to a paleontologist here at Virginia Tech. His name is Scott Evans. He's done some really cool, interesting work on looking at the very first animals that were alive on Earth around 600 million years ago. So stay tuned for that. You'll find a link to his paper down here. And if you like this kind of material, you want more of this kind of content, please subscribe, hit the like button. I'm looking forward to really developing this whole series, and we're going to see where it goes. So thanks for coming. All right, welcome everyone to a conversational series I'm starting, reaching out to scientists from a variety of different fields. Recently, I've been doing a lot more content on evolution, as people have seen. And so, and I saw an excellent sort of news piece that came out through Virginia Tech news on work that was recently published by my guest today, who's Scott Evans. So Scott, thank you for joining me today. I am really happy that he agreed to come in on and talk about his recent work and some of his other work on the larger context of Precambrian fossils and animal life back then. Super interesting stuff that we're learning about. And but let me introduce Scott a little bit. So first of all, Scott, thank you for being here. I really appreciate you coming. Great.
Thank you for having me.
Great. So Scott is actually, I had thought, oh, wow, it would be great to maybe even do this in person. And I was thinking through the technical difficulties of setting up like another camera and another microphone. But Scott, even though he's a Virginia Tech postdoc, he is in Arizona at the moment. So he does his work remotely, right? So so it actually is fine. Like we can just do this over Zoom. As we've learned over the COVID pandemic, we're all now Zoom experts. So thank you for being here and making this work. So let me tell you a little bit about Scott. So Scott, he grew up in New York. He got his bachelor's. He has 2 undergraduate degrees in geology and mathematics from SUNY. And he did a master's and PhD at the University of California, Riverside in the Department of Earth Sciences. Working on Precambrian life. And now he's currently a postdoc for the time being here at Virginia Tech, working in the lab of the Chow lab, right? And continuing some of his work on Precambrian life, looking specifically, I mean, you've done most of your work in Australia, I believe, right? Yes. That's super great. And we're looking forward to hearing that. But I say temporarily, so he's currently a Hokie, and always a Hokie at heart, of course, right? You never can leave Hokie Nation, even though it's only been a couple of years, because you've only been here for a few years. And also just to note, like, you've been incredibly productive. I'm actually super impressed with all the work that you've done. So your first publication was in 2015, I think. So it's like only 7 years, and you have. Well, the CV you sent me has 18 publications, but it doesn't even include your most recent one, I think, which is this PNAS article. So it's at least 19. I guess it's more than that, right? So, and you've published several times in the Proceedings of the National Academy of Sciences, as well as, paleogeology. I believe that's right. Yep. No, paleobiology, several times there. And, really impressive publication record. And of course, big surprise, you've now been hired to be a professor at Florida State University, and that's starting next year, I guess, right?
Yeah, starting in January.
Great.
So staying in the ACC, but just moving a little
right. Well, ACC is a big club, and so it's a fantastic that you've landed that position. And why don't we get into it and we start talking about. Oh, wait, before we do that, I also want to mention that you're an Agouron fellow, right?
Yeah.
Do you want to just briefly say what that is? I think that's super great. That's a pretty prestigious award to get.
Yeah, it's a fantastic program, the Agouron Geobiology Institute, and they have been generous enough to fund 2 years of my research.
That's fantastic.
Thinking about these, the questions I'm going to talk about today, and.
Right.
Yeah, we're supposed to be doing field work in China, and this was awarded in 2020, so that hasn't happened. But we've been lucky enough to pivot and try and produce something of
something. Okay, That's awesome, fantastic. Fortunately for you, like your work necessarily takes you outside. You got to spend a lot of time outdoors. You know, COVID, obviously travel, that there's that, but at least your actual work mostly outside, right? Digging in rocks and
whenever I can.
Yes, that's fantastic. So, if you want to you can start sharing slides if you'd like, and we can start talking about this. It'd be great. So the idea, we're going to talk, give some of the basic introduction to sort of Precambrian life and some of the, if anyone knows anything about, multicellular life, you've heard about the Cambrian Explosion, but the thing that we've learned is that actually it's certainly that is an event, but there's a lot more before that we're learning more and more about. And you've done some really great work on shedding some light on that early life, which is awesome. So looking forward to hearing this.
Yeah, thanks. And
sure.
No, thanks again. It's awesome to have this opportunity to talk with you and your audience about this work that we've been doing. So just jumping in, I put some slides together to try and highlight some of the beautiful fossils we're talking about, and like you can see in this title slide here, a beautiful reconstruction by artist Alex Brosma, who showed some of the changes actually that's going to be a big part of this talk today, and then also just a big part of this paper that we recently had published. And so, as you mentioned, Chris, I imagine that many of people in the audience aren't very familiar with the Ediacara biota. I think if you look at this slide, many of the things, or all of the things that are pictured there, probably don't look all that familiar to most of us. And so I think it's best if we start, before we talk about this paper, which deals with maybe one of the oldest extinctions of animal life. We want to learn about these animals and what they are before we talk about where they went or what happened to them. So the group of organisms that I study is referred to broadly as the Ediacara biota. So I think that's kind of, again, a word that maybe people aren't super familiar with, Ediacara. But it just refers to a group of fossils from a specific period of time. And we'll talk about that exact period of time in a second here. But first and foremost, I think it's just good just to get acquainted with what these things look like.
Right.
So on the left side of this slide, you can see just some examples. These are all actually from Australia, as you mentioned, which is where I've done a lot of my work. And then on the right there is another reconstruction by paleo artist Peter Tressler, hopefully giving you a little bit of a sense for what we think these sort of strange early animals look like.
That's beautiful. Really nice. Now, did you find any of these fossils that are shown here on the left?
I found at least these three on the bottom here.
Cool.
Spent a lot of time digging in the dirt, and I'll show some pictures of that too. But
sure.
That's one of the best parts of my job, is just going out and flipping over a rock and being the first person to see something for over 500 million years.
Right. It's such an exciting thing, isn't it? I mean, I do some on my own, just digging around here, and everywhere I've lived, I've tried to go out and do it. And of course, here around Virginia Tech, we have a rich sort of geological history, spanning around between like 350 million to 450 million years, depending upon where you go. And we can find all kinds of interesting marine fossils in this area, and it's awesome. But also carboniferous plant life, depending upon where you go. And isn't it exciting? Like it's such a rush to like flip something over, and it's the first time this thing has been looked at in literally hundreds of millions of years.
It's so fun. And my partner, she doesn't like going on hikes with me, because I'm especially if we're in a place like around Blacksburg, you're just flipping over every rock and not getting very far. So.
Right. Yes.
So the sort of scientific definition for this biota that we use when we're writing a paper is that it's the oldest group of fossils of complex macroscopic organisms, right? But I think a more intuitive way to think about it is this is really the first time in Earth history that we can actually go out, like we're talking about, flip over a rock And we can actually, without using a microscope, without looking at the chemical components of that rock, we can say, hey, this thing that I can see is a fossil. And what's really cool is that in some cases we can say, not only is this thing a fossil, but it's actually one of our oldest animal relatives, right? So these are really the first animals in the fossil record. So other lines of evidence that suggest animals existed before this time, but these are really the first time we see abundant and really communities of these animals living together.
Very cool.
Yeah, and so they lived between 575 and 538 million years ago, so a little bit older than those things we have in the sort of the Ordovician and Devonian in Virginia.
Right.
But in the terms of the history of Earth as a whole, actually not that old. And so yeah, I like to put this plot of Earth history, like you see on the slide here, thinking about the 4.5 billion year history of Earth, and the fact that simple single-celled things like bacteria have been around for maybe 4 of that 4.5 billion years. Mm-hmm. And then if we look, it's not until around this time, around 600 or 700 million years ago, that we start to see the first multicellular things and really these first animal communities showing up. So really important then, if you're interested at all in how animal life got its start, the Ediacara biota is one of, if not the best resources for understanding that history. So I think there's just so many, and I just started to write down some of these, but major questions that we can ask about animals just by investigating this group of fossils, right? So, what caused this event? What led to the first evolution of animals? What did these first communities look like? You can see, Peter Trussler's reconstruction of what we think those look like. How are they related or not to animals that are around today? And even questions that are really interesting, like what environmental conditions did they live in? And that's really cool because it has implications not just for what happened here on Earth, but one of the things we've been really successful with is thinking about NASA and the search image for complex life elsewhere. If NASA wants to develop that search image, understanding the only place where we do know that complex life showed up is one of the only resources we have for developing that search image.
Right.
Yeah, and then the last question I have here in bold is just the one that we'll talk about more today.
Yeah.
Ultimately, what happened to these guys?
Right.
So yeah, most of them, again, the reason they look so weird is because they went extinct many, many millions of years ago, and so understanding that event, I think, is really important for understanding the whole history of animals on this planet.
Yeah, I mean, it's something that we're learning more and more, the impact of extinction events on the assemblage of the animals that exist at any one time. It's enormous, and we just see it over and over and over again, obviously from molecular evidence, but also the fossil evidence, that it radically changes the kinds of animals that we see. And given that this is the first group of animals, when they go extinct, well, interesting. Yeah, super fascinating stuff.
Yeah.
Right.
Yeah. And we'll see that it's not just fascinating for these animals, but it also is important for animals that we maybe are more familiar with.
Right.
Yeah. So you mentioned that I work a lot in Australia. And so this is a little bit aside from the paper, but I did just want to share because I'm really proud. The site that I've been going to for the last 10 years, where I did my PhD, has actually recently become a national park. And so we're having a grand opening sometime next year. So we've been doing things like building museums. Obviously, I haven't been doing the building, but gotten to help in the planning of these museums. Like you can see in the top there.
The exhibits, you mean? Is that what you're talking about?
Yeah, so places on site where we can actually display these fossils.
Oh, that's awesome.
So that's just really cool to see, from drawings or ideas in our head to now we're starting to finish this up and getting ready for that grand opening.
Cool.
So yeah, and this place that I get. So the Ediacara biota, I should say first, is found all over the world. Every continent except for Antarctica, we've found them, and I'm hopeful that someday someone will find them in Antarctica.
Wow. Well,
But
maybe with global warming, it'll become more and more obvious, I guess.
yeah, one of maybe the only very few positives.
Right, is that we can reveal rocks that are underneath ice. That's, jeez, yeah.
Yeah, I'd rather not.
No, I'd rather not too. But
Yeah.
you got to take, I guess, the pot. If you got to find the sunny side, I suppose that's one thing.
Yeah.
Anyway.
Exactly. No.
Yeah.
And so the other thing, that the reason I put this slide up here is that these fossils that we're looking at are a little bit strange for another reason, which is that they're all soft-bodied. So probably many of you think about fossils as, shells or bones, right? Or even plants, things that have hard, resistant parts. Much easier for those to make it into the fossil record than things that are, again, entirely soft-bodied. And so what that means for us is that we as geologists who want to study this time period and these soft-bodied animals usually have to go to pretty remote, distant places to find them. So again, I've spent a lot of time going to South Australia. I got to go to Yukon this summer, which was an amazing experience, and look for more. So, you can't just go everywhere and find these types of fossils.
Right. That's interesting. So, and the thing is, there's sort of a built-in assumption that, oh, wow, like you could just, if there's billions of years of life on Earth or at least hundreds of millions of years of the time that animals have been around, we should just see like so many different like fossils. But fossilization is a unique event, and the degree to which you're going to have fossilization obviously depends on the kind of animal that it is. And if it's missing hard parts, that's just something that is going to be really difficult to fossilize. Isn't that right?
Exactly. Yeah. So we think about a jellyfish or a worm and trying to get that to leave an imprint or anything in a rock, even for a couple hundred years, is really hard.
Sure.
Much less millions and millions of years. And then the other problem is that these being so old, a lot of things happen to rocks over time. You know? So even if you make it into the fossil record, you could have a mountain building event that completely alters that rock to the point where the fossil is unrecognizable.
Just crumbles apart. Yeah. Sure.
Yeah. Or that, that part of the crust could be destroyed. So it really limits the places we can go.
Sure. I see.
So and when I go to Nilpena and when I'm not doing things like helping put together that museum, the thing I'm doing is probably something that people picture paleontologists, I think, doing, which is we spend a lot of time just digging in the dirt.
Yep.
And trying to find those early fossils. So just put some fun pictures of some work that we've done. One of the things that we do that's a little unique at Nilpena is that we actually find individual layers of rock and we excavate the whole layer because those layers will have hundreds of individual fossils. And it helps us really, you might recognize something right away, or you might come back as we're finding now 10, 15 years later, and you find something totally new. So that's a really cool, sort of different way that we can do this.
So, and then I suppose, because this is going to be essentially like the bottom of the ocean someplace, and this was a layer at one time, and then it was covered over, and then now you have a new layer. And so you want to take each layer off and see what are the animals looking like or other life at that time on that layer, and you can just kind of piece it apart layer by layer. It's almost like a snapshot through time of whatever it looked like at that time. Is that right?
Exactly. And that word snapshot is one we use all the time.
Sure.
And another thing is that a way to think about each one of these layers is that each one represents one storm event that came in and buried the seafloor. And so that's a little unique too in the fossil record. Again, we need unique conditions to preserve these. Right. A lot of times the bones or the shells you find have been, mixed up with a lot of other things over long periods of time. But these specific animals were buried immediately. And so it's almost like time traveling where we can actually go back and look at what the seafloor looked like 555 million years ago.
Now, and then if you have multiple layers like this, and you have another layer on top, and then you guys remove that layer and you evaluate it, and then you have another layer below that. That's what it looks like we're looking at here in the lower left.
Exactly.
So how do you know how much time will maybe have elapsed between the two layers?
Yeah, that's we wish we could tell.
Right.
But we don't. These storms were probably rare. We talked today about 100 or 1,000 year storms as being really big events, right? And we know that these parts of the ocean were otherwise pretty calm to support this animal life, but then we had pretty rare big storm event come in and bury them. So we don't know exactly how long, but probably on that scale of hundreds to thousands of years. So not millions of years, but also we probably don't get every season or every year preserved on these different layers.
You're basically getting one snapshot every couple, maybe at a minimum every couple of 100 years, and maybe even on the order of a couple of 1,000 years even. And right. That's fascinating. Okay.
Yeah.
It's almost like if you were to take a movie and you were able to get one clip, one screenshot at the beginning of the movie and then a screenshot at the end, and now you got to kind of piece together what happened in between.
It'd kind of be like studying the history of humans if you had 40 partial clips from different movies from each decade or something. So we have to do a lot of inferring from what we can.
But fortunately, there's not going to be that much change that occurs between, say, one layer and the next. So you're generally finding, I assume, the same trace fossils and other fossils. Is that right?
Exactly. Okay. Yeah. So we see that the communities that we have about 40 of these excavated beds. At Nilpena alone. And what we find is that we don't see evolutionary change. The same things basically are on each bed. But what we do see that's really interesting is that, one bed will have a completely different, community structure, right? So one organism will dominate one bed, and then you go to the next bed and there's only one of that organism and a completely different organism dominates.
That's fascinating.
One of the things we like to say is if you were able to travel back in time, you would need some fins and some scuba gear, because you wouldn't want to be stuck in one place, but the ocean would probably be very different in different places, like it is today.
Exactly. That's super cool. Okay, cool.
Yeah. So, and the reason we do this, and the reason we spend all this time digging up these layers, is because we want to find as many of these strange animals, as many examples of them as we can. And so, I've just put some examples of the organism that I studied. Basically, my entire PhD was on the fossil that I put on your screen here, Dickinsonia. And so by digging up hundreds of Dickinsonia. I think my total count was about 1,500 different specimens that I had photographed.
Right.
We can start to build a big database of, sort of what is the variation in this organism, and really try to learn more about the biology and ecology of it. So the thing about the Ediacara biota is that they don't really fit nicely into the way we've classified animals today, right? So saying it a bug or a worm or a vertebrate. We can't those types of things.
Right.
So we have to ask these kind of basic questions about them, right? Like, how do they grow? How did they get their food? Could they move around? And in doing so, though, we're able to build a picture of, sort of what these things look like.
Cool.
And so that kind of work has really been the focus of a lot of Ediacara paleontology and paleobiology for the last, 10 to 20 years. There's been a big concerted effort to build these pictures of these individual organisms. But that work, I think, is really what leads us to the paper that brought me here today, right? The paper that we're published, which is, instead of taking that more individualistic approach, to take a step back and think about broader diversity patterns, bigger scale changes that we can build up from those individual understanding of those organisms.
Okay, very cool. Yeah, and this image is just beautiful. So Super impressive. Did you work with the illustrator for this image, or?
Yeah, and yeah, you can see I'm very proud of Alex's work. She did a great job. So yeah, just emailing back and forth. Of course, the
Sure.
virtual world, sending pictures of fossils.
Right.
But yeah, no, this is a beautiful image that Alex created for
It's great.
based on this paper. Yeah, and the reason I put it back here is because what it's actually showing is that in different times in this 40 million year period of the Ediacaran, we see different groups of fossils, right? So I think people are probably very familiar with dinosaurs, right? Different types of dinosaurs: T-Rex, Brontosaurus, Stegosaurus. But if you know a little bit about paleontology, you probably know that they didn't all live at exactly the same time, right? Even though we group them all as dinosaurs, there are different dinosaurs at different periods in Earth's history. And the same is true for the Ediacaran, right? And so we're now learning that there are actually individual periods where different types of organisms existed.
Great.
So just another way to represent that, but here with the names actually shown and the ages where we think these things were around. That we've really split the Ediacara biota into 3 of these distinct assemblages. So yeah, so on the bottom of your screen there is the Agouron assemblage. That's the oldest of these 3 assemblages. And you can see kind of these are dominated by these highly branching, often fractally arranged frond-like organisms. So you can kind of, think of sort of like, I guess, seaweed or sea pens that are around today, standing up in the water column with these high order branching. And then from the Agouron assemblage, the middle assemblage, and this is the assemblage that we get in South Australia. This is the White Sea assemblage. Again, that's the middle of the 2. And I think you can start to see that in this White Sea assemblage, we get an increase in diversity. And we also start to see more than just those fractally arranged fronds. We start to get different body plans and probably a diversification event within this Ediacaran assemblage.
And I don't know if you're going to get to it or get into the details, but it looks like we have an increase in the number of animals that are bilateral, symmetrical, right? So they have bilateral morphology. Is that right? Okay.
Yeah, and this is the time when we see the first, things that we can say for sure are bilaterians.
And so that suggests that they have a front and a back, and they travel.
Yeah, and a left and a right side, and then in most cases we can tell that they can move. So, really important step for things like us who are also bilaterally symmetric.
Right.
And then coming out of that White Sea assemblage, we get into the youngest assemblage, the Nama assemblage, which is interesting because that's actually when we start to see a ramping up of things that are actually making shells. So and starting to actually build maybe some reef type structures. So so just interesting changes that we see throughout these 10 to 15 million year intervals of these defined assemblages.
Very cool.
So so the idea behind this study, I think of kind of as twofold, right? So one is that we have this better understanding of changes through time in the Ediacaran, and we wanted to learn more about that. The other side of that coin is again what we've talked about, which is that when I show pictures that my co-authors sent me to recognize them here, you can see that all the pictures they sent were of them outdoors, hiking, looking at fossils, doing something outside. And that's certainly, I think, something that we all share. And then, of course, in 2020, we had the COVID pandemic, and we were all suddenly stuck inside, not able to travel to places like Australia. And what was really led by my former PhD advisor, Mary Drozer, who's here on the upper left. She said, let's try and build a database of all the occurrences of these Ediacaran fossils around the world, and specifically really build that database with this idea of changes through time in mind. So trying to test some ideas for what might be causing those changes.
Right.
And so that's exactly what we did. Is we built this database. And so, I've listed some of the many characters we tried to collect here. But really the idea behind this was, let's try and collect every piece of information that might tell us more about this Ediacara biota and what's going on. So and so the first thing we did, which maybe is pretty obvious, but I think it is pretty informative, is we just really tallied up the different numbers of fossils that occurred in each one of those 3 distinct time bins. And so you can see, so if you look at the black squares here, those are just showing the total number of genera. One order up from species, in each of these time bins. And what we find is that, the Avalon, that oldest assemblage, has low diversity. And then there's this increase in diversity that we talked about in the White Sea assemblage. And then what we see is a pretty significant drop in diversity from about 70 taxa to about 35.
Wow.
In the Nama assemblage. And then I've put over here the loss of 80% of fossil organisms. Of those 70 taxa that are around in the time of the White Sea, only 14 survive through this through this interval. And so I put the big red arrow on there because that really became the event we wanted to learn more about and try and explore. What's causing this major loss of organisms.
Right.
So there's really, again, we're not the first to recognize this. Others have recognized these changes in diversity through time. And so there are hypotheses that are out there for what might be causing this. And the first idea is that this pattern we're seeing, this drop in diversity, isn't actually reflective of what was happening in the Ediacaran, but it's just really a effect of the imperfections of the fossil record, right?
Sure.
So these White Sea taxa would be around, but they're just not being preserved in that youngest Nama interval. And so as paleontologists, we often talk about this as preservation bias. But I think Mark Laflemme and authors in 2013, they cleverly termed this the Cheshire cat model, right? This idea that, those fossils are there, they're just invisible to us today because we're not seeing them for whatever reason. Mm-hmm. And again, this database that we constructed allowed us to test that. And so this is figure 2 from our paper, and it's showing some of the different preservational things that we looked for in this global Ediacaran record. Mm. So just there's a lot of information on this slide, I understand, but just to the things to hone in on here. First, on the left side there, we know that the continents don't just stay in one place. So these are maps of what the continents would have looked like, or what we think where they would have been in Earth's past during the times of these 3 distinct assemblages. Yeah. And then we plotted up where we find these fossils on those maps to see where these organisms basically were living. And so what we see is that in the middle here, in that time of the White Sea assemblage, our localities are all within low latitudes, between about 0 and 30 degrees. Mm-hmm. And that's kind of interesting because we know that today high diversity is found, preferentially near the tropics. And mm-hmm. so that's one possible explanation for what's going on here. But then when we look at our fossil localities from the Nama assemblage, what we see is that we actually have a lot of our localities that are falling within that same range.
Yeah,
right?
I see that. Yeah, only a few exceptions that are outside of the range. Yeah.
Yeah. And so that just is telling us that if high diversity taxa are limited to these low latitudes, we should still be sampling the right places to see them.
Sure.
Even though we're not. Another thing we can look at is the different places basically in the ocean that are represented, the paleo environments.
Right.
That are represented from these different localities. And I think without getting into it too deeply, the takeaway here is that these pie charts for the White Sea, the high diversity assemblage, and the Nama, that low diversity latest Ediacaran assemblage, they're very similar. Right?
Yeah, yeah.
Again, we're probably not missing some environment in that latest Ediacaran interval.
Right. So basically, if the Nama assemblage, for instance, if all of a sudden, really all you had access to was just, say, slope and deep basin rocks, well, then that would be a bad sign that, like, okay, well, we are missing some of these other, sea environments. But you have virtually the same percentages. And so, right, then that should be, you're going to have a range of animals that should be, potentially adapted for those different kinds of environments, right?
Exactly. Yeah. If those high diversity White Sea assemblage fossils are there, we're sampling the same environments. Right. So we should see them.
Right.
And then the last thing here is showing preservational mode, the different ways that fossils or organisms make it into the fossil record. And what we see here is actually the opposite of what we would expect in that Cheshire cat model, right? Mm-hmm. So instead of losing ways to preserve fossils through this interval, we're actually picking up more and more different ways to preserve fossils. So again, if these guys are around, they should be making it into the fossil record.
Yeah, that makes sense. Like, for instance, for your Nama assemblage, for the preservation mode, imagine if that was the White Sea assemblage that you saw. That you had that wide range. You had pyrite and carbonaceous compression and cast mold carbonate and the silica based. If you had that range in white assemblage, well, okay, you have also a diverse range of organisms. Perhaps it's because of the type of preservation mode why it's diverse. But in fact, you have the opposite. You have more different kinds of preservation modes, yet you're seeing fewer fossils.
Exactly,
or fewer species, I should say. Yes.
Yeah. Right. And yeah, exactly. With more preservational modes, we should actually expect our diversity to increase rather than decrease, like what we're seeing.
That makes sense. Great.
Yeah. And so when we combine all of these different things, really what we're seeing is that what we have from the fossil record doesn't support these imperfections, this preservational bias being responsible for this drop in diversity, and that this looks like, as best we can tell, a real extinction event. This looks like we really did lose many, many different types of organisms across this White Sea to Nama interval.
Great.
And so from there, then we can start to test other hypotheses for what might drive the loss of those taxa. And so the first hypothesis that I put up here is this idea of biotic replacement. So the basic idea here is that a new animal or many new animals or certain type of animal could show up and essentially be incompatible with these Ediacara biota taxa, or specifically these White Sea taxa. And I always struggled to come up with the right image for this. But I went with a watering hole because, there are a lot of different animals that rely on watering holes as an essential resource.
Right.
And if we imagine some animal evolving that is better at getting water from that watering hole, or maybe more importantly, was consuming all of the water to the exclusion of these other animals, then that might lead to the extinction of certain organisms, right? This idea that competition for a particular resource can maybe drive extinction of certain groups. And so that's the idea we tried to test by looking at different ecologies that are present in these different time bins, as well as looking at specifically at what goes extinct. And one of the key things we would expect is that there should be specific types of ecologies that go extinct preferentially at this interval that are impacted by whatever new animal is showing up that's causing their extinction, right? Animals or things that we don't see in this picture that aren't reliant on the watering hole, they wouldn't be impacted by that hypothetical new thing that removes the watering hole, right? So we should see some type of selectivity in our extinction.
Okay.
So so again, we turned to that database, and that's exactly what we did. So I won't go into all of the different things we looked at, but one of the things that I think is kind of intuitive is thinking about feeding modes, right? If there's something that came along that was better adapted at taking a resource that you feed on, you might go extinct.
Mm-hmm.
And so I'm showing 4 different types of feeding that we have in the Ediacaran. And the real thing here, I think, to focus on is the stack bar histogram on the left. And what we can see is that middle white sea assemblage looks pretty similar in terms of the relative abundance of these different types of feeding modes that are around. So we're not seeing some shift to a new feeding mode, but we're seeing the same relative percentages in these 2 distinct time bins.
Can you talk a little bit about how you know the feeding mode of these animals?
Yeah, that's a great question. So it's based on whatever information we can find.
Mm-hmm.
So a lot of it is actually based on seeing trace fossils of these different animals. So right, so the things I've shown you have all been body fossils, but we also know that animals leave behind traces when they move specifically, and sometimes when they feed. So actually, this image here in the top right part of your screen is an organism we described a couple of years ago called Ediacara, and it leaves behind these traces in the mat where it actually consumes this organic matter that's sitting on the seafloor as it moves.
Mm-hmm.
And so it's from those trace fossils that we can tell it's consuming that organic matter.
Okay, so how do you know that?
Just the reason that it leaves those trace fossils is that it's actually disturbing and removing that organic mat layer.
Okay, so it's not just being displaced, like, okay, it's being disturbed and then sort of removed, and right, like, because if this is like a muddy bottom or something, or it's probably more than mud. There's probably all kinds of organic material that it's eating in this mat.
Yeah, so basically, in the Ediacaran, there aren't many things that actually go down into the sediment and mix the sediment up.
Right.
And so what happened was these layers of organic material actually formed on the seafloor. They covered the seafloor.
Mm-hmm.
And so, we think about this as like a layer of pond scum, essentially, that lined the seafloor.
I see.
And so it turns out that was a really great resource for the things that could actually adapt to consume it.
Sure. And then obviously things that are like frond shape, that would be suspension feeding. Is that right?
Yep.
Yes. Okay.
Yeah. So things that sit there and don't move don't have any other way of feeding, essentially.
Right.
And then seeing those adaptations to stick up into the water column, to expand. To grab as many food particles out of the water as they can.
And it looks like, so mat feeders like Dickinsonia, which you've done an enormous amount of work on. You had another PNAS paper about Dickinsonia, I believe, a couple of years back. So what it looks like here is that they likely are moving and traveling and then leaving depressions in the mat. So you can see that as well. Sometimes you have the animal itself, and sometimes you have a depression of the animal. Is that right?
Yeah. So Ediacara on the top here, it moves through. This is probably more like what we think of sort of worms or things moving through the sediment and consuming as they go. Dickinsonia looks like it doesn't have a mouth or an anus. It doesn't have apparently any openings, but it just seems to have sat there and absorbed. It might have had symbionts or released some chemical that allowed it to dissolve the mat. But the takeaway is that it leaves behind, these Dickinsonia-shaped depressions in that mat.
Wow.
And so we can actually see trails where we have these multiple depressions, and then they end in a body fossil of Dickinsonia.
And then that's why it's probably so important to get these full layers out, so you can kind of track that thing over multiple chunks. If you were just removing all the chunks and not putting the jigsaw puzzle together, or at least pulling out the entire jigsaw puzzle together, then you wouldn't be able to follow these traces or like track a particular Dickinsonian, trace, at least these mat depressions over a few places.
Yeah.
Is that right?
And actually, one of the beds we excavated, we did just that. We flipped over a rock that was sticking out from the side of a hill, and we found one of these trace fossils. And yeah, we just kept digging and digging to try to find the Dickinsonia, and it spanned about 3 meters.
Wow.
So
even though it's pretty small, we're talking about, these Dickinsonians, based on the scale, you were showing them just a couple of inches basically across, right?
Yeah. So yeah, a couple inches. The biggest ones we have are
some are really big. Yeah, like you had one that
really big,
that looked like it was a 10 centimeter scale, so that would mean that it was at least maybe half a meter. Is that right?
Yeah, it's exactly half a meter. Yep.
Jeez.
Yeah. So 2 feet, and that's really rare. And I'll tell you, when we found that specimen, we did some hooting and hollering on the hillside. That was really fun. But yeah, most Dickinsonia are a couple of inches is a good average size.
That's wild. Okay.
But they could move pretty far,
sure.
which is really cool.
Yeah, that's super cool. Okay.
Yeah, and so taking all of this information we have about different feeding modes in the Ediacaran, right? We can also look at, and I think this is maybe more intuitive than the stack bar histogram I showed earlier. This is showing the top pie chart here, all of the things that are around in the White Sea and the different ways they fed. So all 70 of those taxa, and then below that we're seeing what goes extinct. Mm-hmm. And I think the point here is that these pie charts look essentially identical.
Yeah, they look very similar. Right?
Yep. So we're not seeing, specifically these things that feed on the mat going extinct, or specifically things that are suspension feeding going extinct. So there's no selectivity in terms of feeding mode for this extinction.
Wow.
And then, if we dive in, if you wanted to read more, certainly you can dive into the paper and the supplement.
Yep. And there'll be a link to the paper, obviously, in this video.
Great.
Yep.
Yeah. Mm-hmm. But the takeaway when we look at a bunch of different ecological characters is that there really doesn't seem to be much selectivity of this event in terms of those distinct ecologies. So really from this, and this is figure 3, again, from our paper, the thing to notice is that these White Sea and Nama stack bars look pretty similar across these intervals. And I'll also add, I didn't put it because it's pretty boring in a PowerPoint, but we also tested this statistically to make sure, we weren't just eyeballing.
Right. Yep.
But we don't get significant change between those events, with one exception, and that's the last stack bar here, surface area relative to volume.
Oh, I see that. So yeah, relatively low surface area to volume ratio in the, at least, the percent of animals that had low surface area to volume ratio in, I see. Okay.
Yeah. So I'll get into this more in a couple of slides.
Right.
So what's cool is that sort of broad range of impacted ecologies is actually consistent with another hypothesis for what might have been going on here, which is that we had some major environmental change that was detrimental to a variety of different organisms that were around at the time. So again, I've sort of crudely pasted the picture of the asteroid that killed the dinosaurs, because I think that's an example that everyone's familiar with. No evidence for an asteroid impact causing our extinction event. Mm-hmm. But the idea here is that there are these major abiotic factors, these environmental factors that can cause significant change in the Earth system, and that can lead to, and often do lead to, these major extinction events. And so the thing, though, that if we want to be sure that this event is due to environmental change, we really would like to link a specific type of environmental change to the taxa that we see disappearing. And that's exactly what happens when we look at this metric, surface area relative to volume. So just a sort of primer for why we might care about surface area to volume. I'm trying to illustrate in these cartoons here, which is, things like those Avalon assemblage fronds that I showed you that have those complex branching patterns, right? They're sticking up into the water column, and they're really maximizing surface area to volume. And in doing so, that means that a bunch of their cells, a high proportion of their cells, are in direct contact with seawater. And so the reason that's important for our story is that if we have low oxygen in our environment, that is better adapted to get oxygen to its different cells than something that doesn't maintain a lot of those cells in direct contact with seawater, right? Things that have multiple thick tissue layers and are usually filled with metabolic fluid that's not in equilibrium with seawater, right? They need things like a blood vascular system to get oxygen to those different cells. And just in general, they're not as well adapted to lower oxygen conditions, right?
Sure.
So we looked at this because it's a potential proxy from the paleontological side for oxygen tolerance. And as you pointed out, that middle White Sea assemblage really contains a high number of taxa. About half of the taxa that we have during this time show really none of these morphological adaptations to maximize surface area relative to volume, right? So these are sort of dense, solid things compared to the things we see a lot in the Agouron assemblage and in the Nama assemblage that have these really complex branching patterns and do a bunch of other things, have really thin tissues that allow them to maximize that surface area relative to volume. And then the thing that I think was really informative to me and really hit home this story is looking at actually the things that make it through this event, the things that are around in that White Sea interval that actually survive into the Nama. And so that's what I'm showing here in these two pie charts, right? The top is all 70 of those taxa that are around again.
Right.
And then you can see in the bottom, the things that actually survive, the 14 taxa that make it through. And 13 of them are these high surface area to volume taxa. And so that's really suggesting that we're seeing preferential survival of organisms that are adapted to better survive in low oxygen conditions.
So the percent, I'm trying to do the math in my head here, the percent of those taxa that were high surface area during White Sea that also survived into Nama, it's relatively high. It's probably what, like 80% or something like that made it through. I'm trying to think of the numbers, because you say it's you have 70 taxa or so in White Sea. So the top pie chart is about 70, and then below you have 14. And so, oh, maybe it's less than 80%. So maybe like 50% or something.
Yeah, something like half of the high surface area to volume taxa survive, whereas basically all of the low surface area to volume
except for one of them. Yeah.
Except for one go extinct.
Right.
Yeah.
Wow. Okay.
So it's a pretty significant pattern.
Yes, very significant pattern. Yeah.
For such low numbers.
Right. You're seeing, and this is true across. So for this assemblage, the White Sea assemblage, we're talking about locations across the entire world, right? That we're collecting these from.
Yeah. So we can look in Russia and Australia. And Canada, and see the same patterns, the same things.
So White Sea assemblage, you're gonna have this ratio, probably about half or so are gonna be these low surface area to volume animals, but then you're now looking a few tens of millions of years later, not even that much, maybe just 10 million years later. Now all of a sudden, almost all of those animals are completely gone, and all you see are just these high surface area to volume animals. Across the world. Yeah.
Across the world. Yeah. Right. And despite the fact, again, that you have the same preservational conditions, the same environments, the same locations.
So something happened. Yeah.
Yeah. So something happened. And I think the really intuitive thing, and the thing that makes, I guess, me comfortable as a paleontologist is that other geologists, geochemists who study chemical compositions of rocks can show what's happening with oxygen levels. And it's difficult to tie those records directly to our paleontological records because we use different types of rocks. But the takeaway that almost every geochemist from this time will tell you is that oxygen was fluctuating a lot during this time.
Right.
And so we know oxygen is something that's fluctuating a lot, and we're seeing a response in the animals that suggests that they're responding to a drop in oxygen. And so I think we get really nice multiple lines of evidence that support this same conclusion.
Correct. And so I know your paper doesn't cover that, or at least it discusses it, but you don't have those data in this paper, right? Is there data out there, like other studies showing that, like for the Nama assemblage, that there is this drop in oxygen, or is it like you say that there just happens to be these massive shifts that seem to occur around this time?
Yeah, so there are papers that do suggest that there's low oxygen
During Nama. Okay.
And this is like anything in science, it's highly debated.
Of course. So, yeah.
So there's different theories, and again, the really tricky thing is linking these different records up, right?
Right.
So saying that that drop in oxygen happened exactly when this extinction happened is difficult, but I think from the paleo record we can say that, well, it looks like globally these things are responding in a way that suggests that reduced oxygen. So I think that's becoming a more and more powerful story.
Great.
So yeah, so that's, I mean, those are the big things we talked about in this paper. So going back to looking at these different faunal assemblages, I think the key story is that a lot of these and some of my favorite things that are in this middle white sea panel, probably don't make it into this NAMA panel because, again, of that reduction in global oxygen availability, and really a big environmental shift seems to be the most likely explanation for what's happening here.
Great. So yeah, go ahead. No, go ahead. Sorry.
Well, I was just going to end with thinking about this sort of in a broader picture of, what happens to animal life after this. So that there's one thing that I think is really nice about this story is that despite the fact that I've talked a lot about how weird and different these animals are, what we're seeing here is the same thing we see in animals for the next 500 million years. So this figure on the right here is one that shows a number of marine families through time. And what's highlighted with these arrows are the big 5 major mass extinctions that paleontologists often refer to, right? These are big events where we see major drops in diversity. And in fact, comparable drops in diversity to what we saw in the Ediacaran.
Right.
And so the one that killed the dinosaurs is the end Cretaceous here. And we know that was likely caused by this large meteorite impact. And then there's other major drivers, I guess, for these other extinction events. But one of the things that links them all up is that they're all the result of they all caused major climate change, right? Whether it was a volcanic eruption or a meteorite impact, it caused major perturbations to the climate system.
Yeah.
And so I think that's consistent with what we're finding in the Ediacaran.
Right.
That this was the result of we have a major change in climate, and we see a major loss of most of the things that are around.
So let's talk a little bit about that. So for some of these things, like you pointed out, we've got the meteor impact for the end of the Cretaceous. If you correct me if I'm wrong, but I think right now we think the Permian-Triassic boundary is largely driven by like massive volcanic eruptions that led to massive disruptions of the overall environment, and that was even worse than the Cretaceous extinction. I mean, we lost more than 95% of all diversity at that point, right? Yeah. So what do you think is then the potential cause of this change in oxygen that drove the loss of diversity for the Ediacaran?
Yeah, I wish I had a good answer for that.
Okay.
But short answer is I don't.
Sure.
But one of the things that seems that's possible, I guess, is that we have major tectonic events, major movements of the continents that can often lead to major changes in things like ocean circulation or and can really drive these low oxygen events. Right. It could be things like volcanic eruption or a meteorite impact. We can't rule those out.
Sure.
But I think the general idea is that oxygen is so is lower than it was today, and is so variable at this time that it could be that a small change really led to this drop in oxygen.
Yeah, especially since it's so selective, right? You have the the high surface area to volume ratio animals mostly making it through just fine, and the loss of the low surface area to volume ratio animals. So, yeah, I think right. That makes a lot of sense to me that it seems to be a selective, all of a sudden there's a somewhat of a significant disruption to the environment, and unfortunately these animals that had evolved this particular body plan was perfectly suited for high oxygen environment, but now all of a sudden there's some change to just change oxygen levels, and unfortunately they're not suited, so natural selection just gives them the boot, doesn't it? So at least it's consistent with that. Yeah. So there are other let's see, since we're on this topic again, correct me if I'm wrong, but I believe there's some speculation that either it's the end of the Ordovician or the end of the Devonian, that some of that was, and I think it's the Devonian, that there may have been a massive increase in oxygen that could have been responsible for an extinction event because of potentially a biotic contribution that like onset of photosynthesis. Correct me if I'm wrong, because that's not true, because photosynthesis likely evolved several, maybe at least 1 billion years ago, I believe, right? Right. But that there was this massive increase in oxygen that occurred at some point.
So
I might have that wrong.
No. So one of the things that happens around the time of the late Devonian, just before that, we see the first evidence for plants.
Yes.
And so they're a big photosynthesizer.
Right.
And the Devonian is when we start to actually see the first sort of forests and establish, we get the first big trees from places like upstate New York, where I grew up.
Mm-hmm.
We start to see, 30-foot tall trees in the fossil record. So big, massive forests.
And leading to the preservation of carbon, which is why we have the Carboniferous. Carboniferous means it's bearing carbon, right? So, and we didn't, and what we intuit from that is that the organisms that are responsible for decomposition of this massive plant material really hadn't come on board yet. So life was very different back then, where you had significant preservation of plant material leading to carbon, right? Isn't that right? And coal.
Yeah, and I think the idea is that the advent of land plants likely fundamentally changed the carbon cycle, right? We know that's a big part of the carbon cycle today, and so that's gonna have major impacts on the carbon cycle, but then also the oxygen cycle is linked to that as well, and so it caused these major fluctuations that are also probably responsible for these,
and ultimately impacting animals in the ocean, which is what you're showing here.
Right. So it's and again, I think one of the things I've been asked a lot about this study is, what does it tell us about modern climate change? And I think a big lesson from all of these events is not necessarily the specifics, but that when you start to turn these knobs in any one way, they have far-reaching impacts for a lot of different systems. And so letting those knobs work naturally as best we can is a really good idea.
So, and then Nama, that takes us right up to the beginning of the Cambrian, isn't that right?
Yeah.
And so do you think that this extinction event could have been, I mean, you might be speculating here. I'm asking you to speculate, I guess. But do you think that this is potentially sort of how we see, as you point out, we got the age of mammals, and of course birds had an enormous expansion in post-Cretaceous, age of reptiles after the end of the Triassic. We often have a rapid expansion of a variety post-extinction. Do you think that that's a possibility here, that there may have been some kind of event, disrupting the assemblage, the biota that was in the Precambrian environments, and then opening up niches once things got there, and then that's maybe what explains the Cambrian explosion?
Yeah, no, it's exactly right. And I'll add that, yes, we are speculating a lot here, and there's a lot more work that needs to be done to prove that. But I think, one of the significant, hypotheses that comes out of this study is that, we could see that sort of resetting of ecosystem that ultimately, does away with these very weird early animals and allows animals that look much more like modern animals to show up during that big that big Cambrian explosion. So no, yeah, that's exactly right. And the other thing too that I find kind of interesting is that it may be that those few low surface area to volume things that survived then had to be really adaptable to survive in this time of low oxygen, maybe that spanned, through into the Cambrian. And so it may be that adapting to those different variable oxygen conditions could have also promoted innovation and also, helped us get to more modern type animals.
Sure. Excuse me. Okay. So another question on that. One thing that we see with the Cambrian fauna is complex interactions between species. We have the, things like Ediacara with like predation that's occurring. Is there any evidence at all of that kind of interaction between animals in the Precambrian? Like, was there a predator or anything like that?
So there are certain shelled organisms, things that do produce hard parts that do show borings in the Precambrian.
Oh, okay.
But largely what we see is that there's no evidence from, like, when we excavate those beds in Australia, we don't think anything was eating anything else.
Right.
I guess with the exception of things like Dickinsonia eating that organic matter that's just sitting on the seafloor.
And the organic matter, there's probably quite a bit of it that is alive, but it's likely single cell organisms and that kind of thing that it's just munching on or absorbing, I guess.
Yeah, but probably another, a reason that so many soft-bodied things were so happy just sitting out on the seafloor is that there weren't those big predators like Anomalocaris that came along and could consume them. So so it's a totally wacky time, but a really interesting time for the start of animal life.
Do you mind talking a little bit about how you got into fossils? Like, when did this become? Obviously, you're very passionate about it. So when did this become a passion for you?
So I grew up in upstate New York, and my driveway, sort of like Virginia, had a bunch of Devonian and Ordovician marine fossils in it and little fool's gold pyrite. And so, I started a fossil collection, I think in kindergarten or first grade, just searching through my gravel driveway right outside my door. And so, since then, I've always loved paleontology. And then went to high school and became too cool for it a little bit. Didn't know what I wanted to do going into college, so I started as a math major, and then just took a general course in paleontology and geology as a freshman, and then haven't looked back since. So,
great.
It's been really fun.
That's amazing. Do you want to talk a little bit about what you will be doing when you go to Florida? I assume continuing with this work, or do you have other, other projects lined up that going in some radically different direction? Or there's a lot. There's still a lot, obviously, to answer about this. So,
So we're still. I'll still be working in Australia.
Great.
So that's my list of projects, it continues to grow from there. We have some new taxa that we've discovered recently that hopefully we'll be publishing very soon here.
Cool.
And that's always really exciting.
Right.
Then I'm also, I'm planning to try and go to the Yukon. I got to go this last summer. And those are places that you need a helicopter to get to usually.
Oh, wow.
So even more remote than in South Australia, and just, so places where not that many people have ever been. And where we're actually finding even more of these organisms and really testing the idea that we have here, right? If we go to a new place, we have a new opportunity to see, hey, do these things still go extinct at the same time, or are we totally wrong? And just continuing that type of work wherever I can.
Right. Fascinating stuff. I really appreciate you giving us time to learn a little bit about what you're working on. Really interesting, important work. And, I think that, you're studying right at the edge of what we know, just as all science, science should be, right? We know so little about animal life during this time, and you're obviously putting the pieces together, and we're learning more and more about it. And it's obviously under very challenging. Well, at least the fossil preservation part. I'm sure you probably have hot days out there in Australia, and sometimes it's physically challenging to do this work too. But it's so awesome that you do this, and I really appreciate you sharing this with us.
Yeah, no, thank you. And I guess that's a great transition to the last slide, which is just to thank all the different funding bodies that
Yes.
allow me to do this challenging, but yeah, as we talked about at the top, really fun to be there and yeah, unearth something that hasn't been seen the light of day for 550 million years. So yeah, definitely all of the
Great.
work I showed wouldn't be possible without yeah, these
All
different
the
institutes.
support. Yeah, awesome. I really appreciate you doing this. And yeah, thanks for coming on. We'll be definitely following your work. Hopefully, if it's some new discovery, happy to mention it, and maybe bring you back on again to talk more.
Yeah, fantastic.
Great.
I'd love to.
Great.